In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more: the creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene